Hello everyone, welcome back for yet more human physiology. So last time we talked about a lot of the organelles in the cell with a particular emphasis on the endomembrane system. So basically all the organelles involved in the synthesis of large macromolecules like proteins and lipids, and then how do we transport them and package them up and send them out all over the place. So the focus of this video is specifically going to be on protein synthesis. So I mentioned in the previous video how very important proteins are. Proteins basically mediate all of our physiological processes. So all the different physiological processes that we're going to talk about throughout the semester, whether it's the nervous system, the heart beating, the pancreas regulating your blood glucose, everything is mediated by proteins. So understanding how a cell goes about making a protein in the correct way is going to be essential. So let's get into it. So first, let's make sure that we understand kind of the basic strategy that the cell is going to follow here. This is what's called the central dogma of molecular biology. It basically describes kind of the progression of how the cell uses DNA to eventually make a protein. So there's kind of a lot to unpack here. So if we start at the top of this picture here, you see DNA replication. So this is not, we're not quite as interested in DNA replication. So this is the process of a cell that is about to go through the process of mitosis, replicating its chromosomes, making exact copies of the DNA, so that we can split those chromosomes up equally into two daughter cells. For protein synthesis, we're not really interested in that. So the process that we're going to be interested in are twofold: transcription and translation. So transcription is going to be the process of you start with your DNA, particularly a very specific part of the DNA called a gene, which we will define later, and we are going to transcribe that gene into a short messenger RNA transcript. And then translation, once we have made that messenger RNA, it is going to be exported out of the nucleus through one of those nuclear pores into the cytoplasm or into the rough endoplasmic reticulum, where the messenger RNA will be read in a certain way by a ribosome. The ribosome will read the messenger RNA and use the instructions in that mRNA to put amino acids together in a particular order so that we get a functional protein. So there are some disconnects here. These are some things that we kind of need to understand about why the process has to be this way. One thing you might be thinking is, well, why can't the protein just be built directly by using the DNA as the blueprint? So here's kind of the definition of the gene that we need to know. A gene is a segment of DNA that provides the instructions for making one protein. So basically think of kind of like blueprints for a house. Picture that there's some warehouse in your city, whether it's Tulsa or somewhere else. Imagine that there's a big warehouse in which stores all the blueprints for everyone's houses that lives in Tulsa, Oklahoma. Well, your blueprints are somewhere in there, your neighbor's blueprints are somewhere in there, the blueprints of all the people who live far away from you in some other part of Tulsa are in there as well. Each one of those blueprints is one gene, but all the blueprints being held together, that is the chromosome. There are many, many, many genes on a particular chromosome. One gene provides the instructions for building one particular protein. So what you might be wondering is, okay, if the DNA, the gene, is the instructions for building one protein, why do we need messenger RNA? Why can't we just use those blueprints to build a protein directly? Why do we kind of have to have that middleman there? Well, there's two reasons why we have to kind of have that step in between the DNA and the protein. There's two reasons why we need messenger RNA. The first issue is that the DNA, the chromosomes, are in the nucleus. They're always going to be in the nucleus. So, as we said, a gene is just a small part of a whole chromosome. The chromosome isn't going anywhere. We can't really freely transport the chromosome anywhere. It's stuck inside the nucleus. The ribosomes, those little machines that are going to be making proteins, they are outside the nucleus. They are either in the cytoplasm or embedded in the rough endoplasmic reticulum. So, in this way, there's not really a way to get the builders and the blueprints in the same location, right? So this is what messenger RNA is going to do for us. Basically, we make a little copy of the DNA. It's not a whole chromosome, it's just a short little copy of that particular gene, and we can export it out of the nucleus and get it to the builder. We can get that messenger RNA to the ribosome, whereas we couldn't get the whole chromosome to the ribosome, right? And then even if that wasn't the issue, here's the other problem. Ribosomes cannot read the DNA because, as we said at the end of chapter 2, DNA contains the nucleotide thymine. Ribosomes can't read thymine. It's basically not a letter in their alphabet. What they can read is uracil, and guess what we know about RNA? RNA replaces thymine, which is usually found in DNA, with uracil. So RNA is readable by the ribosome, but DNA is not. So these two issues that we bring up here are completely solvable by just having the process of transcription, making a copy or a transcript of that particular DNA gene, making the messenger RNA. We can get the messenger RNA out of the nucleus to the ribosome, and then once we do that, the ribosome can read it. So as I was mentioning, any particular chromosome could have as many as hundreds, if not thousands, of genes on that particular chromosome. But as I was saying, one particular gene is the instructions for making one chromosome. And as such, one messenger RNA contains the information for that one particular gene. Now we can make many copies of that messenger RNA so that we can make a lot of protein really fast, but the idea here is that one messenger RNA contains the information from one gene. So the way this works is that an enzyme in the nucleus called RNA polymerase, when we want transcription to occur, it will bind to a part of the DNA that is further up past the gene, which is called an upstream region called the promoter. So it binds to this promoter region that is upstream of the gene, and then it starts moving downstream until it gets to the gene, and once it gets there, it starts making RNA, using the DNA as a template. And you'll notice here that in green here, this RNA transcript, we're making a complementary set of RNA. The only difference is that we're replacing T's with U's, as we said. So once the transcript is finished, so this whole process of transcription is taking place inside the nucleus, once we have made our transcript, once we've made our messenger RNA, we export it out of the nucleus through one of those nuclear pores that we were talking about in the previous video. And once we export it into either the cytoplasm or the rough ER, that transcript is going to be captured by a ribosome, that big structure that is made of both protein and ribosomal RNA. So what the ribosome is going to do is literally start reading the message. So it's going to start at one end and start scanning across. And what it's doing is it's looking for a start sequence. So basically a little message that says this is where you start building the protein. When it first starts reading, it basically is a whole bunch of gobbledygook until it gets to that start sequence. And then if it starts reading from there, everything should kind of start to make sense and it should start building the right protein. So when a ribosome is reading the messenger RNA, what the messenger RNA really contains instructions for is which amino acids are we using and in which order do we put them together. So basically what the ribosome is doing is it contains the enzymatic activity we need to form peptide bonds between amino acids. So basically all those dehydration synthesis reactions we need to build a protein and to make a long polypeptide, all those reactions are catalyzed by the ribosome itself. But here's the problem. Here's the problem with the messenger RNA transcript. There are 20 different amino acids, as we learned at the end of chapter 2, but there's only 4 RNA nucleotides. So there's clearly no way that the messenger RNA gets read one nucleotide at a time. You would basically have to have some way of, for every one nucleotide, being able to determine which 5 amino acids that means, and there's no way to do that. So how do we solve that problem? Well, the way we do that is the ribosome actually doesn't read one nucleotide at a time. It reads 3 nucleotides at a time. So each run of 3 consecutive nucleotides is called a codon, and this actually allows us to account for all 20 amino acids. With 4 different nucleotides, there are actually 64 different ways they can be put together into stretches of 3 that we call codons, and obviously that's more than enough to code for our 20 amino acids. So the extra 44 there accounts for a lot of redundancies and some start and stop sequences, as we're going to see here. So the mapping, the codons, and the specific amino acids they code for is sometimes called the genetic code, which is something that you might have heard of before. So this chart here gives us the entire genetic code. And I'll go ahead and warn you, I definitely would not wait to kind of get this whole thing memorized. It's going to take a long time, but you definitely want to get this thing memorized. Now, I'm just kidding. You don't need to memorize this. So hopefully your heartbeat didn't go up too fast there. Yeah, so do not memorize the genetic code. I don't have the genetic code memorized, so you don't need to worry about that. So memorizing the whole genetic code is totally unnecessary for us, but there are a few things that we should kind of take note of here. So remember how I said when the ribosome binds the messenger RNA, it's looking for a start signal? Well, that start signal is down here. It is this codon AUG. So basically, when the ribosome is reading the transcript, it's looking for a stretch of nucleotides that goes AUG. So that codes for the amino acid methionine, which is why almost every protein in humans starts with that particular amino acid. And then all the other codons that you see here, you can tell if you, say, for example, have 3 uracils in a row, that means the amino acid phenylalanine. If you have adenine, cytosine, adenine, that's going to code for the amino acid threonine. So with these 64 different combinations of nucleotides put into codons, we can account for all 20 of our amino acids, except for 3 cases. You'll notice UAA, UAG, and UGA. Those are all what are called stop codons. These are just as important as the start codon. The start codon tells the ribosome when to start putting amino acids together, and the stop codon, as the name suggests, is going to tell the ribosome to stop. Without a stop codon, the ribosome would keep reading and reading and reading through the messenger RNA transcript, and it would make a protein that is far longer than we really intend. So the start codon tells us when to start making the protein, and the stop codon tells us exactly when it's time to stop. So if we want a little more information about the ribosome itself, we mentioned this before, but it's basically this big amalgamation of protein and ribosomal RNA. So that ribosomal RNA is made in the nucleus, and then the ribosome is basically put together out in the cytoplasm. So the best way to kind of think about this is the ribosome is basically this really big enzyme that catalyzes the formation of peptide bonds that we need to make a big protein, right? However, there's one last issue that we kind of have to clear up here. We know that the ribosome reads the transcript, and that determines which amino acids we're going to use, but how do we kind of make that connection there? How do we say, okay, I read this as a start codon, AUG, how do I get that methionine specifically in there? How does the ribosome know that this codon means we want this particular amino acid? So this is the adapter that we're suggesting we need here, and these adapters are called transfer RNA or tRNA. tRNAs attach themselves to a particular amino acid, and tRNAs have a particular sequence of nucleotides here, which is called an anticodon. An anticodon, just like a codon, consists of a run of 3 nucleotides, except these are going to be complementary to the codons that we find in the messenger RNA. So, for example, if you look at AUG here, which is the start codon, the transfer RNA that carries the specific amino acid we want for that methionine is going to have an anticodon that matches up with AUG, so it is complementary. So the ribosome scans the messenger RNA, and when it comes across a particular codon, the ribosome will simultaneously bring in that particular transfer RNA. The anticodon will bind to the codon, and the ribosome will take that amino acid and use it to start building a protein, as you can see in these pictures here. So we've gone into quite a lot of detail, it seems, for a physiology class, right? In terms of how do we build a protein. Well, why the heck do we care about that, right? It doesn't seem like it would have anything to do with human physiology, right? Well, here's why we want to care about this. Here's why we kind of want to know about this. I've already mentioned that proteins basically do all the heavy lifting in physiological processes, and we've already talked about several examples of important proteins, right? We've already talked about the sodium-potassium pump, insulin, both insulin and the insulin receptor, and how important they are in glucose homeostasis. We've already talked about what goes wrong in the synthesis of the CFTR chloride channel in people with cystic fibrosis. Collagen and keratin. For collagen, we talked about why vitamin C is necessary to build the protein collagen. So we're never going to be short on examples of why protein synthesis is extremely important. So thankfully, we didn't get into all the excruciating details about protein synthesis, but I do think it's kind of important that you at least have a good understanding of the fundamentals behind how proteins get made. So, to end off our discussion on protein synthesis, let's again, as we suggested with cystic fibrosis, let's talk about how protein synthesis can go very wrong. So one of the most common ways we can screw up making a protein is if there's a problem with the blueprints for making that protein. So if the DNA represents the blueprints that tell you how to make a protein, then a mutation would be kind of like an error or a typo in the blueprints. So imagine the blueprints for your house. So let's say that you live in a really nice brick house. Well, what if someone in the blueprints before your house was made crossed out the instructions that say to build out of brick, and instead they say build out of sand or dirt or something? Well, that's going to be a huge problem. It doesn't seem like a huge issue, but if you're changing the instructions on how to build something, then the whole structure is going to be completely different. It's not going to be as we intended, and certainly the function and stability is not going to be the same. So again, if the DNA is the blueprints for how we build a protein, a mutation is a change to those blueprints. We are essentially either changing which amino acid we stick where, we shorten the protein, we make the protein too long. There are a number of different ways we can change the blueprints for how we build proteins. And if we change the blueprints significantly enough, then the protein is not going to function correctly, as we saw in cystic fibrosis. So what you're seeing here in this diagram are what are called point mutations, meaning a single nucleotide change that can change how the protein is made. So, for example, on the far left here, if the DNA blueprints are supposed to be TTC, the transcript should read AAG, and according to the genetic code, that should code for the amino acid lysine, which is very hydrophilic. A silent mutation is, if you're interested in maintaining good function in your protein, this is the best kind of mutation you can get. We change a nucleotide, but because of redundancies in the genetic code, that change in nucleotide does not produce a change in the amino acid. So the DNA changed, but the protein did not. One of the worst types of mutations you can get is what is called a nonsense mutation, in which a nucleotide changes you from a codon that codes for an amino acid to a stop codon, meaning that the protein ends up being way too short. The ribosome stops building the protein way before it's supposed to. A missense mutation is one that is fairly common, in which a nucleotide change causes a change in which amino acid we use. So there are two different types of this one that can occur. A conservative mutation is one in which we change the amino acid, but we change from one amino acid to a very similar amino acid. So if we start with lysine, lysine and arginine are two very, very, very similar amino acids. So you're probably not going to suffer too much just from changing from one amino acid to one that's very similar. A non-conservative mutation is a much more serious one. This is where you are changing from one type of amino acid to a very different amino acid. So maybe you're changing from a hydrophilic amino acid to a hydrophobic amino acid. And if you do that, that's going to totally change how the protein folds, because it's having to bury an extra hydrophobic amino acid, or it's going to have to fold in a particular extremely different way. So that's going to cause a lot of different problems. Other types of mutations that can occur are insertions, in which we insert a big segment of DNA into a gene. A deletion, we delete a big part of a gene. Those are common too, but thankfully not super duper common. So some examples in which mutations can cause certain diseases just because proteins did not get made correctly. A very prominent example is sickle cell anemia. So this is an example of a point mutation, a non-conservative point mutation, a missense mutation in a gene called beta globin. So beta globin is one of the proteins that makes up hemoglobin. So what is supposed to be in that beta globin protein is the amino acid glutamate, but because of the mutation, we instead replace that hydrophilic glutamate amino acid with a hydrophobic one called valine. So this causes the beta globin gene to not fold correctly, and it causes hemoglobin to start tangling up into these long fibers, and that causes the red blood cells to kind of form that telltale sickle shape. The example that we already looked at is cystic fibrosis. So this is a nonsense mutation. So this causes the ribosome to stop making the protein far before it should. So the chloride transporter doesn't end up folding correctly, and it gets destroyed by the lysosome. And we already talked about kind of what else happens with cystic fibrosis. So diseases like this that are caused by these mutations to the DNA, which cause proteins to not function correctly, these are called genetic diseases, and we are not going to be short on examples as we go throughout the semester. All right, so that is going to do it for our discussion on protein synthesis. So the very last thing that we're going to want to cover in this chapter should be a fairly short discussion. We're just going to get some very general information on the cell cycle and chromosomes. So we are not going to do the whole cell cycle. We're not going to do prophase, metaphase, anaphase, and telophase, and all that, so do not worry about that. That's more of a general biology discussion. But we are going to talk a little bit about cells dividing and the importance of stem cells. So I hope you will join me for that discussion. I will see you later. Bye-bye.